I think we're going to go ahead and get started. So good morning, everyone. This is the next in the series of the Viruses and Vaccines seminar. My name is Ellen Chu, and I'm delighted today to have Trevor introducing Trevor at the beginning, going over a monkeypox genomics update, and then subsequently we'll have Anna Weil and Leah Starita speaking to us about the University of Washington Husky Coronavirus Testing program. So Anna is a close colleague of mine. She is an assistant professor in the Division of Allergy and Infectious Diseases. Her research focuses primarily on studying how the gut microbiome impacts susceptibility to enteric infections. But for the last 2 years now, she's also been heavily involved as the co-PI of the Husky Coronavirus Testing program here at the University of Washington as part of the UW COVID-19 response. Dr. Leah Starita is assistant professor in genome sciences and co-director of the Brotman Beatty Advanced Technology Lab, the BAT Lab, at the University of Washington. And she works on genomic technologies to solve problems in public health. And for the last 4 years now has led the lab portion of the Seattle flu study, and in fact was the first person to design a working molecular assay for SARS-CoV-2 in the United States and helped us identify early transmission. So I'm delighted to have all three of you here today to speak to us, and I'll turn it over to Trevor.
Hi, everyone. So just going to share screen, and I'm going to keep this brief, setting a timer. So, okay, so monkeypox has actually been very interesting with what's going on, and so I thought I'd kind of share a small update here rather than talk more about SARS, at least for this month. So, so if we have a monkeypox virus genome, it's a DNA virus rather than an RNA virus, and so because of this, it has a much larger genome. This is cowpox because I couldn't find monkeypox, but it's actually quite similar. We have 200,000 letters, bases, rather than the normal 30,000 that we have for something like SARS, SARS-CoV-2, or 13,000 that we have for influenza. And because of this, it can fit a lot more genes into this genome. So we have all these kind of accessory genes, unlike many of the RNA viruses. And because it's DNA, it has much higher replication fidelity. And so we might generally expect one mutation per genome per year along a transmission chain, as opposed to the SARS-CoV-2, one mutation every 2 weeks along a transmission chain, just as kind of our prior expectation. If we look at monkeypox viral diversity, historically, there's been classified as these two clades, West African clade showing up in Ivory Coast and Nigeria, and a Congo Basin clade showing up around the DRC. We've been working to kind of change this nomenclature a bit, and we're now referring to things as clade 1, 2, and 3, where 1 is around the DRC, 2 is around Ivory Coast, and 3 is around Nigeria. And this diversity has been built up over many years in the animal reservoir, which is actually thought to be rodents, not monkeys. Monkeys were just where it first was noticed. And we have, like MERS or some other viruses, these stuttering chains where it kind of will frequently spill over from the animal population into humans, but doesn't really historically transmit well between humans. And so these outbreaks have been historically self-limiting. However, if we look at the global monkeypox outbreak right now, we can see that things look very different. All of these viruses are very closely related to each other, showing up primarily in Europe, but also elsewhere. And they all descend from these clade 3 viruses, and then showing up also similar, descending from these viruses in clade 3 that were sampled from Nigeria and the UK, Israel, and Singapore in 2018. If we look at these 2018 viruses versus these 2022 viruses, so 2018 viruses versus 2022 viruses, we see a bunch more evolution than we might have expected for a DNA virus. So we have 47 single nucleotide changes compared to 2018. This is one mutation per month rather than one mutation per year, as we would have expected. So just this is a big surprise. And if you look at what these mutations are, this was first noticed by Andrew Rambo, where you have the if we just look at these mutations, they're almost all GA to AA. So I'm changing G to A here in this GA to AA context, or TC to TT, which is just the same thing in reverse complement. And so this is this hallmark of editing by ApoBec. So it's a host enzyme that's designed to kind of protect against things like RNA viruses by mutating them to death. However, this seems to be basically not being, working partly well, and just instead of kind of completely killing off the virus, it's actually really resorting at a higher mutation rate. And what we're seeing there is that this additional ApoBec editing didn't appear to be happening in the animal reservoir. So we look at historical evolution, everything looks normal. There aren't the kind of special spectrum of mutations. But once we hit 2018 here, and along all of the 47 changes leading into the current outbreak, kind of ApoBec signal is very strong. And even within the outbreak, we're seeing a fair amount of evolution, and then that fits this ApoBec signal. So we have a pretty consistent story, although it is a pretty surprising hypothesis that monkeypox jumped into humans in late 2017 and has been circulating in humans since late 2017. We can see this because of the change in mutation spectrum. And then it just didn't kind of catch very well. So it's kind of percolating along for these almost 4 years. And then it gets into a particular population that sexual transmission kind of really facilitates transmission of monkeypox. And then we have a kind of a large outbreak that's associated with that, not jump into humans, but then movement into a population that facilitates transmission. So that is what I think is going on. And I guess we'll learn more as more sequenced. And yeah, and so we can conclude human-to-human transmission has been occurring since late 2017. And I will stop there and hand it over so we can get to the main show.
Thanks, Trevor. I'll just go ahead and get set up. Oops, Robin, I can't share, it looks like.
One participant can share at a time. Trevor stopped sharing.
While you're working on that, just for everybody else, we're going to hold all questions until the end. And then if you want to go ahead and start populating the chat, we'll go through them after Anna and Leah are done.
Can you try now, Anna?
It says host disabled participant screen sharing.
Okay.
It looks like Anna needs to be promoted to co-host.
I think you got bumped, Anna. My apologies.
Okay. I'll just start talking while we're sorting it out.
There you go. Try again.
Oh, there we go. Great. Okay. So I am excited to be here. Can you guys see that? It's okay.
Except it's in presenter view.
Oh, it is. Let me switch it. All right. How's that?
Great.
Okay. So thank you, Helen and Trevor and others for the invitation to talk today about the Husky Coronavirus Testing Program. This has been a really mammoth project that was put together initially on a short timeline as the University of Washington asked Helen to set up a testing program for the university at a time when testing was hard to come by. So in summer of 2020. So I'll be talking today about some of the challenges that we encountered and also about how we solved some of the problems of scale, some of the results as well. So moving along to an overview of the project, which I'll give first before moving into some of those challenges. The Husky Coronavirus Testing Program is testing a SARS-CoV-2 testing program for all members of the UW community who have an active net ID. All participant contact, including results reporting, enrollment, and daily attestations are conducted through text or email. So the daily attestation is one of the hallmarks of the study, which is a communication that occurs every day. Most people choose to receive this by text that asks people if they have symptoms, if they've had recent exposures, and other questions. And this is how we base a lot of our testing off of these responses. Testing mechanisms are observed or unobserved collection with PCR that Leah is going to talk about more. We have testing sites 7 days a week on the upper campus, and also some unobserved testing sites at UW Bothell and Tacoma, as well as the Seattle campus. And data sharing, since the beginning, we've been working in very close coordination with the UW President's Office and the public health arm of the university, the EH&S, Environmental Health and Safety Group, as well as the county and state. So I wanted to just show an example of our primary testing area that we've been using for the last 2 years. This is UW Club, which was repurposed as a testing site in 2020. This shown here on the right is Anne-Marie Couchy visiting one of our, the registration table at the front, and then the testing sites are a little bit back further, and this is the original swab we were using. This image is from the original marketing done by the university for the program. So a very big one-slide overview of results and enrollment is I'm going to talk about next. So currently there are actually, I think, 39,300 and something participants. Every day we still have people newly enrolling. These are primarily students. There have been 224,000 tests performed in the last almost 2 years. Around 6,500 positive tests, around 3,500 genomes sequenced. And I mentioned the daily attestation as our main area of communication. There are 3,000 to 4,000 of these 39,000 participants that actually fill this out every single day since they've enrolled. So we have a huge amount of data on symptoms and travel and other questions we've asked about over time from kind of this core group that we think of as a core surveillance group who fill this out every day. And in January, we began collecting data on rapid test results as well. Even though there is no rapid test testing in our study, we ask participants about if they've taken rapid tests. We're also now accumulating data on this front. So I want to talk a little bit about year one and how we started out, because this was really in the fall quarter, the pre-vaccine era. And it was during this time that we encountered a lot of our scale issues that Leah and I will both be talking about more. So at this time, campus was online only, although many students were still living, especially in the Greek community and in some dorms, and there were no vaccines available. So we originally tried to stratify groups by what we felt were higher risk for infection. So students living essentially in residence halls or dorms, and then also stratifying them by whether or not they came to campus. We ended up testing and sending invitations for testing for people when they enrolled in the study, when they reported symptoms, when they reported an exposure to a person who was infected, or if they had recently attended a high-risk gathering, which was defined at the time as an indoor gathering without masks. We also planned to conduct surveillance, and we had hoped to also be able to launch outbreak testing when needed. So this study design was quickly shifted from surveillance to testing of the Greek community in the fall based on outbreaks in that population. Fortunately, we had a good rapport and relationship with this group already from the testing that had been done over the summer. And we shifted really to testing these members of the study every 3 days for the beginning of fall quarter, and most of our infections during this time were in the Greek community. But we concluded from this that if we were going to be able to scale this to more groups, especially when people came on campus in the future, that we needed to run more tests. So with that, I'll turn it over to Leah, but I'll be advancing slides for her.
Thanks, Anna. So, right, so not only did we have to scale up, but we immediately had a bigger problem. So right after the Husky program launched, we got a phone call from Roche. So underneath this little is extraction necessary sign is a Roche extractor that extracts the nucleic acids for qPCR testing. And they called my lab manager and said that they would no longer be supplying us with extraction reagents. So we were in trouble. So we had to do something. And we were worried that this was really going to affect the launch of Husky testing and the ability to continue. So we had already switched to those lovely mass-produced Q-tip looking swabs that you could see that we were allowed to ship dry, which was great, or use or transport dry. And because we could transport them dry, we thought, well, if we could elute these swabs, elute the specimen off these swabs in a PCR-friendly buffer, did we really need or did we really need hardcore RNA extraction for these samples? And so that's the hypothesis that we were going with to see if we could develop an extraction-free test. Go ahead, slide. Oops. Yeah, so we tested, the first thing we did was we tested a panel of buffers and detergents and decided that plain old Tris EDTA or TE was the best elution buffer for avoiding PCR inhibition, which was great because it's practically free and we can make buckets of it for very little amounts of money. So we added a digestion step, so where we treat the samples with proteinase K. And we did this because we noticed that we were having some particular problems with snotty samples. So we tried to digest the mucins. And then we heat treat the samples to 95 degrees for 15 minutes to inactivate the virus, which is another thing that extraction provides you. So we wanted to be able to handle these samples safely. So that was really important to us. And we also do it's a quite long heat treatment, about 15 minutes. And this actually helps stabilize the RNA and also now kill this protease that we had added to the samples. And this was actually spearheaded by a graduate student that we stole from Jay Shinduri and Cole Trapnell in the spring of 2020. And a bunch of people from my lab. So obviously we have to make sure that this test is going to be work well. And the first thing we did was we pulled 30 known positive samples back out of the freezer and looked to see if we could again detect SARS-CoV-2 with just this proteinase K and heat treatment. And when we compare to the crappy extractor that we had bought to replace the Roche, you can see that the extraction-free or Swab Express, as we called it, actually identified more samples, SARS-CoV-2 in more of those samples that were known positives. And then comparing across actually about 1,100 samples, it compared exactly. So comparing extraction to extraction-free side by side, we found that the extraction-free or Swab Express was 100% sensitive at identifying samples with SARS-CoV-2, 99.9% specific. So there was one sample where the Swab Express actually identified SARS-CoV-2, whereas the extraction didn't, and a very low failure rate, because this was something we were worried about, and it's only about 0.1%. So we were worried that stuff you find in swabs like boogers would be inhibitory, and it turns out that we really weren't seeing that at all. And we have the difference in CT values between extracted and non-extracted is actually really low, under 1 cycle for our ORF1b target and about 1.5 for spike. And again, our tests all have a human control in them because they're unobserved. We want to make sure people are sticking these swabs in their actual noses. So we can see here that that the RNase-P is also detected at pretty much similar levels. Go ahead, Anna. Okay. So important to the future of Husky testing, we reduced the per test reagent cost by nearly $5. And we could process more tests in parallel in the 2 $600 ovens we bought on Amazon.com compared to processing in serial on a single extraction robot. I'm not going to name which robot this is, but it's not a very good one. But still, it costs a lot of money, and now it's actually sitting gathering dust on a shelf, which is awesome. So time to about 384 samples. So you'd have to run the machine 4 times in a row is about 6 hours on the extractor, whereas we can just throw as many plates as we want into our cheap little ovens. And we gained a lot of efficiency and obviously cost reduction doing this. Go ahead. Okay, so that was for year 1. For year 2, we wanted to further optimize to allow for flexible scaling, because we knew that there were going to be some events like move-in testing, for example, where they were going to want us to test a lot of people all at once. And the way we approached that was we turned to these rhinostic swabs, or we call them tiny swabs. And they're a small swab that's attached to the cap of this tiny little tube. These tubes are, when they're racked like this, are compatible with a standard 96 well automation, lab automation. And there's a lot of other great things about these swabs. They have 2D barcodes on the bottom. They're really handy. Go ahead. So there's pros, right? So lab automation compatible. They were available, which a lot of people on this call know is like more than half the battle. And you know, they're tiny, right? So we can store them, store thousands of them without, you know, having boxes lined all the hallways. But there are worries as well. So, you know, they're tiny. Are we gonna have space for enough labels? Are the participants going to be able to use them, right? They're so small. Here, if anybody's watching, I can show you one right here. They're so small that we worried about dexterity and also test performance, right? So this is a hard plastic swab versus an absorbent Q-tip like thing. So were these going to work as well? Were they going to collect a good specimen? And then also potential contamination. And I'll talk more about that in a minute. Go ahead. Okay, so the first thing we did was we recruited 69 individuals from infants to 65 years old to participate in a usability study for implementing these swabs. And when we asked if the swab was easy to handle, 80% of people or so agreed, which was great. Nobody strongly disagreed, which was important. And 97% of people also felt confident that they had completed the nasal swab correctly, right? Or they had collected a good specimen. And this was not surprising, right? This is the summer of 2021. People were used to swabbing themselves at this point. But the thing that did get us was that they are hard to see. These labels are hard to see. And, you know, there's just not a lot of room to, you know, increase the font on this. This isn't as big of a problem with Husky testing. The demographic we target with Husky testing is, you know, 18 to 22, most of the participants, so they can see just fine. But with other studies, we actually send them a cheapo magnifying glass to read the labels. So that was good to see that people could use them, and we could kind of help fix this vision problem. Go ahead. Okay, and you can see here there's just a tiny little label on the side. Go ahead. But it turns out that people were collecting good samples with good specimens with this. So in comparison to the typical anterior nares swab on the left, there was, you know, perfectly respectable amounts of RNase-P on these swabs, and we lost about 2 CT values. So, you know, this was not worrisome that we were going to start failing a bunch of samples. And go ahead. And the limit of detection was nearly identical to the regular anterior nares swabs as well, which is, you know, tens of thousands times better than the rapid antigen tests. Go ahead. Okay, so this is where we were worried about cross-contamination. Anna, can you play the video? This is a little video of our 96 well decapper in action. And you can see that the swabs are kind of hanging over the tubes at the end. And when when Michael Springer from Harvard first showed me these swabs, I said, no way, they're gonna drip. They're gonna be a contamination issue. But you know, they published their paper, it looked great. Next, yeah, and we did a whole bunch of checkerboard experiments where we put, you know, samples with something in them next to, you know, surrounded by something without, and we did not see, you know, any contamination. We don't see SARS-CoV-2, you know, happening positives next to each other, which is really important. Go ahead. So we were happy with that. And the most important thing is that we gained a ton of efficiency here. So it takes one technician more than 3 hours to elute 1 PCR plate's worth of samples using standard swabs, but they can accession 94 swabs in 34 minutes, which is a 7-fold improvement. And so it's really helped us gain efficiency. So with, you know, fewer staff, which also helps to keep our prices down. So with our extraction-free protocol and our new automation-friendly swabs, we were able to provide testing at a scale for the whole university, even through massive surges like Anna will tell you about next.
Thanks, Leah. All right, I'm going to move on to talk a little bit about our results from the pre-vaccine era very quickly in fall quarter. We found not a lot of positives overall compared to what was coming, but most of them were in students. The majority of them were in people who actually did not have symptoms. Our overall test positivity was low. The highest predictors of positivity were contact with a known COVID case, report of COVID-like symptoms, and also people who tended to walk in without an appointment for testing, people who didn't report to us any reason for testing. When they came in, they had a higher percent positivity. And with some of these people who didn't report symptoms, when we followed up with them after their test and talked to them about symptoms later on, we were able to identify a large number of people who were pre-symptomatic at the time of testing. Often these were people who were exposed to a roommate, and we were able to identify that the CT values in those pre-symptomatic people were higher than symptomatic people. So we also found during that time that the percent positivity overall was lower on UW campus than in the surrounding King County. This was likely due to an increased availability of testing during a time when testing was harder to come by. But we also hope that this high availability of testing allowed the outbreaks that did occur on campus to be somewhat contained. You can see at the bottom, this is the Greek community who had most of their infections in the beginning. The red line of faculty and staff actually had very few infections until kind of this holiday time and an outbreak in a group of maintenance workers. And then this bigger curve is overall test positivity in King County. So we wanted to look into this further regarding our thoughts about if testing had helped to contain some of these infections, or if there had been any spillover of infections from the UW community into King County. This is a divergence plot of SARS-CoV-2 genomes made by Amanda Casto, showing in green the Greek cases. You can see this group is identical genomes, so very closely related transmission. In the blue, the community genomes. These are from publicly available genomes from testing done in King County at the same time. And then in yellow, a few non-Greek cases also from HCT. So what you can see from this is that the Greek cases are very closely related, that the other positives from on campus are actually not related to the Greek community and are more closely related to other transmission going on in King County. But most importantly, we saw that these Greek outbreaks, and this is just an example of a few other instances as well, there were not, forward in time or in genomic variation, there were not cases going into the community from these Greek outbreaks. So we were glad to see that and hope that this availability of testing maybe contributed to containing some of those outbreaks. So big picture lessons learned and areas of scaling up. We did work a lot with the University of Washington regarding balancing priorities of on-campus spread and preventing that versus greater good testing. I would say, testing for anyone who wanted it. We found that people who walk in for testing for no reason do come for a reason. So we wanted to make sure going forward that we had availability of testing for people who didn't make appointments and who just showed up randomly for testing. We found that outbreaks can derail our plans. And we, probably our biggest challenge was that our data usage was really beyond the scope of what was possible with UW REDCap at the time. And this caused outages of, daily attestations and testing invitations. And we corrected this by buying more servers. And this really helped immensely going forward. So this is just a figure from one of our meetings with them in which they showed us that when our study launched, the REDCap usage more than doubled at UW. So that was not, serving other studies well that were also crashing during this time. So that was one of our major issues. So during year 2, we encountered a different campus. It was open. There was also a mask mandate as well as a vaccine mandate. So we had now a fully vaccinated population by January. We continued to test people on enrollment in the study. And when they returned to campus, as Leah mentioned, there were giant testing pushes of thousands and thousands of samples a day when people returned to campus, especially moving into the dorms. We continued to invite people for tests when they reported symptoms or exposure. We got rid of our high-risk gathering question and swapped it out for traveling out of state or out of the country, consistent with CDC guidelines. We continue to conduct outbreak testing much of the year with weekly testing in people who live in congregate living settings. And then we launched a new testing mechanism called drop boxes, which is a testing station that is manned by a staff member so that questions can be answered and kits are given out either by appointment or by walking in. People can then register that kit using a QR code, so it gets connected to their name and their net ID, and then they can drop off the kit anytime after swabbing, and the kit contains directions for them as well. So we used, I think, around 17,000 of those kits during the return to campus event, for example. But we had an expectation overall during the second year that most testing would really be by walk-in. So when I think of year two, it's hard to escape really just thinking about the first 2 weeks of January, which was a time when the study was really tested on all fronts. And I'm happy and proud to say that really no one was turned away from testing at any time during this period. And it was a time when in King County and even on campus, it was hard to find testing elsewhere. So during this time, and thanks to Leah's innovation, also in the lab with many others, we were collecting 1,500 samples a day for a while. This only goes until February, but there's really been consistent testing, around 600 to 800 tests a day gathered up until the end of spring quarter as Omicron continued to evolve in this population. The gray here is total collected, and then the colors are the sequence samples. So one of the areas that allowed us to succeed during the Omicron surge was the utility of drop boxes, because this mechanism of testing requires fewer staff. You don't need someone to register the test, guide someone through doing the test. And a lot of these people now, they didn't need guidance doing the test. They could read the directions and they had done it lots of times. So you can see here on the left, the time scale isn't here, but this is during move-in where we use those thousands of Dropbox kits for testing. People did continue using the drop boxes a little bit. This is among students, but during Omicron, when there were very long lines at UW, we actually opened a new drop box in the middle of, I think, this first week, thanks to Devin McDonald and Tessa Wright, who managed the drop boxes and the UW Club kiosk. And people use them a lot during Omicron, and then they continue to use them during the rest of the year. This goes through June. So we found that these drop boxes were, people found them convenient and faster than standing in line at UW Club. And we also found the same in faculty and staff who really didn't use them at all during the fall. And then during Omicron became used to them and then continued to use them through the rest of the year. So based on this, we did decide to emphasize drop boxes more in our year 3 plans, which I'll talk about. So from a genomic perspective, we found that Omicron infections were very closely related, but it was harder to identify clusters in this time period because there was just so many infections at the same time. This figure again is by Amanda Casto. But essentially in this highly vaccinated population, we found that there was explosive spread even in the setting of maximum use of NPIs. So our lessons learned in year 2, I would say we improved our ability to collect data about walk-in people who are testing for no reason by requiring them to fill out the daily attestation when they registered a Dropbox and reminding them if they're standing in line at the kiosk. We did have some people pick up kits for the Dropbox and then swab themselves and drop them off without registering the kit with the QR code. So we didn't know whose kit that was. That was a bigger problem at the beginning, and people became very used to this quickly. And now we have, I think, less than 2% unregistered kits coming back. Really, one of the main challenges in January was also quarantine and isolation of staff across all areas of the study, which we got through by grad students, and really anyone who was around was working at the kiosks. Also, the new Dropbox that we opened really helped alleviate the unprecedented demand we had for testing on upper campus especially, but really our investments in the year 1 laboratory innovation, data management and programming, and these low staff testing mechanisms, the Dropboxes, this all really paid off at a time when testing ramped up so much so fast, and we were able to meet that need, fortunately. So next, I'm just going to go through a couple of slides of data from year 2, and then a few areas that were our new areas of research. So in January, we began looking at household transmission of Delta compared to Omicron. On the left is our Delta households, where we had groups of people who were becoming infected in the same short time period. The blue is the time of symptom onset and red is the positive result. At the beginning, when we started looking at this, we only had a few Omicron households, but we noticed early on that these serial intervals between symptom onset were shorter. And as the Omicron cases accumulated, it actually became hard to know if people were being infected in a co-primary transmission event outside of the household, since people who live in the same households also do the same things, or if they were really being infected in the household. So Kyle was looking into this and teamed up with Amanda Casto again to look at genomes within these households. And by looking at the huge number of samples we had and identifying only households that had identical viral genomes, so people we know transmitted most likely to one another, and also who had symptom onset on different days, we were able to really narrow down this serial interval with much more confidence using identical viral genomes, showing here the difference between the serial interval and Delta much longer here than Omicron. We also looked into the time since last mRNA dose for people who became infected during Omicron and compared this to Delta. This figure I'm just going to go through a little bit more. On the left-hand side here is the date that the last dose was received, and this figure only has vaccinated people and only has mRNA vaccinated people. So, for example, people here who received their last dose, if you take them forward through the graph, you'll see that their infection occurred. This is a Delta infection in blue in September of 2021. So their first positive is shown on the x-axis here. So there were some boosters available at the time of many of these Delta infections, especially in November and December, but you don't see any boosted people becoming infected with Delta, which are the open circles shown here. And the gray, I should mention, is the booster eligible pool. And I would say the main thing you can see from this figure is when people became infected in our study. So the Delta wave is shown here and Omicron here. But even as more talk of Omicron was occurring and people went to get their booster in November, October, November, December, they still, many of them, at least in this graph, went on to develop infection in January and February. So we've removed people from the 14-day period after their dose, but overall there's a fair number of infections you can see here among boosted people, and then many who were unboosted at the time of infection. With Trevor Bedford's lab and in this work led by Marlon Figgins, we also looked into the reproductive number between Omicron and Delta. On the top here shown is just the epi curve of the variant cases in for Delta and Omicron, and then the variant proportion in the HCT population. The switch from 100% Delta to 100% Omicron was extremely fast. It was much faster than we saw in King County. And then on the bottom here, the reproductive number estimates with confidence intervals shown here, comparing Omicron in orange with a much higher peak reproductive number. And this kind of fanning out here at the end as new variants were coming on at the end of February as BA2 was increasing. These are a few new areas we're looking into, is in this large population of people who are healthy and active in the community and doing things on campus. How many of them are developing reinfection? I'm just going to go through this figure also, since it's a little bit complicated. So on the left, we have the date of their first positive swab. So if you look at maybe, let's say February 2022, if pretend this line is one person, this person tested negative. These are all blue dots of testing negative. And then here, 1 year later, on February 1st of 2022, they tested positive. So all of these test positive people, again, showing that the waves of infection, Delta here in the fall of 2021, and then Omicron here in February, but really continuing on through May. So this figure goes until, I think, May 15th. And what you can see is that among people who tested negative all along and then were infected first in either the Omicron wave or earlier on with Delta, many of them did go on to have another infection, at least another one or maybe two infections. And these are by positive PCRs after 90 days in May of 2022 or in February if they had an earlier infection. So in these groups, we're really trying to eventually accumulate enough reinfections to understand more about the risk factors for this, the CT values and other characteristics of reinfection, including with genome sequencing. But one thing you can see easily on this plot is that before people had a positive test, they tested a lot and frequently with all these negatives. And then after they became infected the first time, they're really not testing as often. And many more of these were actually people who were symptomatic. So we did have many people with symptoms multiple times that were tested. And then my last data slide here is our new data on rapid test results. As I mentioned, we added this to the daily attestation in January. And among especially that group who communicates, those thousands of people who do fill out their daily attestations, we have gotten a fair amount of rapid test data from people every day, including percent positivity. This is on June 16th of around 11 or 12%. So we are hoping that over time we can understand more about why people are using rapid tests, what are they experiencing when they use them, and what are the reasons for these maybe invalid results or when they don't know the results. We also look into these participants to see if they had a test with HCT, a PCR test within 7 days of their rapid test, whether it was negative or positive. And we did find above 90% concordance between those results. And we also want to look more into those people who are testing positive by rapid test and negative by PCR, what the characteristics are of the reasons for their tests, what their CT values are, and other questions about rapid test use, since this is now a much more common mechanism of testing. And lastly, we do continue to track variants of concern going forward. Now, this is up until June 1st with BA5 and BA4 coming on the scene, but still at this moment on UW campus, there is still a lot of BA2 and BA2.12.1. So moving into year 3, my last slide, I'll just mention first that the planning for year 3 was really informed by the activities and preferences of our study population, what people found convenient, and also by the University of Washington priorities going forward. And their plans are that there are not going to be mask mandates, and that they're also going to be providing antigen tests for large testing events such as the return to campus testing. The PCR testing through HCT will continue for symptomatic people, exposed people, and those who want to confirm rapid test results. We are also continuing PCR testing for people who come and ask for it, even if we don't have a reason that they would need it. And this is in the spirit of always supporting those people who come for walk-in testing, knowing that this is a high-risk group. We're transitioning fully to a Dropbox-based testing mechanisms, and we'll probably use pop-up kiosks for surges. And research objectives going forward include some of the topics I mentioned, but also vaccine efficacy, long COVID symptoms in students and other populations on UW, vaccine beliefs and practices over time, and more about rapid tests. So with that, I will thank, there are so many people that work on this study, particularly undergrads who staff much of the lab and all of the kiosks and drop boxes. And that is a group that really did an amazing job during the Omicron surge and really worked extra hours and made sure that everyone was getting the tests that they need. Also to thank Helen for her leadership in this study and the rest of the Seattle flu study PIs. And then the people who make the day-to-day run are shown here: Jess O'Hanlon, Julia Bennett, Tessa Wright, Devin McDonald, Caitlin Wolf, Kyle Lutien, and Natalie Lowe. And then I don't know if Leah wants to take over any of these acknowledgements or say anything specific, but so many other people in the BAT lab, programming, running lab tests, making sure return of results is running well, and then many others at UW in the data management sphere and at EHS. And we're happy to answer any questions.
Great, thanks. That was great. It was really wonderful to hear all the work that's been done both on the clinical side, on the lab side over the last 2 years as we plan for year 3. It looks like many of the questions around monkeypox have been addressed. Trevor, are there any other things that you wanted to respond to?
No, I think chat handled it. If anyone, Cecile, Nita, and folks, if you want to get in contact, please go ahead.
And then in terms of questions around the Husky testing project, there's one question related to whether or not we're measuring immunity, COVID immunity, and whether that might be relevant in this population.
We are not measuring immunity, meaning I'm assuming serologically or with blood sampling. We do not have any aspect of the study measuring that area, but I will say there are a number of other studies that Husky Coronavirus Testing program participants have enrolled in through the University of Washington. So many people do go on to participate in additional studies. Helen, you might have some other things to add about that.
Yeah, I think one of the things that we do is that when people do test positive within Husky testing, especially with the novel variants, they are offered enrollment into other studies. And this is what feeds into a lot of the work that's done by Jesse Bloom and Marion Pepper and David Wiesler, who spoke at earlier seminars about identifying the immune response to the variants and then to hybrid immunity. So. Any other questions? Okay, great. Well, thanks everyone for coming today. And Trevor, did you want to go ahead and introduce the next speaker?
Yeah. So next month on July 19th, same time, we'll have Hannah Alteen from the YDUH speaking. So Hannah has been very involved in kind of the genomic epi side for on YDUH. I'm not exactly sure what the seminar will be on though. Yeah, and really great to have everyone attending today.